Hi everyone, my name is Jenny Kuan, and today I'll be presenting my project on consensus stocking using various computational tools and targets. This project was conducted under the supervision of Dr. Matthew Shapira at the University of Toronto. Drug discovery marks the first step in the drug development pipeline and plays an important role in identifying novel therapeutic targets. The process can be divided into two main stages, discovery and development. The discovery stage involves identifying targets and compounds, establishing biological activity, and selecting clinical candidates. The development stage focuses on clinical trials, which assesses for safety, efficacy, and therapeutic potential in humans. Traditional drug discovery is often time-consuming, labor-intensive, and expensive, typically taking 10 to 15 years and costing over 1 to 2 billion USD dollars. One major challenge is the high failure rate in clinical trials, which can reach up to 90%. These inefficiencies highlight the need for a more streamlined and cost-effective approach. One approach is computer-aided drug discovery, or CAD, which uses computational algorithms to analyze biological and chemical data, as well as predict how drug molecules interact with target proteins. CAD can significantly reduce both the time and costs associated with drug development. A key component of CAD is virtual screening, an in silico technique used to identify small molecules that are likely to bind to a target protein. There are two main types, ligand-based virtual screening, LBVS, and structure-based virtual screening, SBVS. Ligand-based virtual screening uses the structures and pharmacological profiles of known ligands without requiring the 3D structure of the target protein. In contrast, structure-based virtual screening relies on the protein's 3D structure to screen compounds for binding to its active site. For this project, we will focus on structure-based virtual screening. A typical workflow includes target selection, protein and ligand preparation, identifying the binding site, performing molecular docking, and calculating binding scores. These scores will predict how well a ligand fits into the target's binding pocket using mathematical scoring functions. While different docking programs may generate similar binding poses, their predicted scores often vary, and there's no definite way for determining the correct binding pose. This variability can lead to false positive, lowering the hit rate of virtual screening and making it harder to identify truly active compounds. In fact, using a single docking program has been shown to have notable limitations. To address this issue, consensus docking has emerged as a promising strategy, as research has shown that combining results from multiple docking programs can improve the overall accuracy of predictions. Consensus docking leverages the strengths of multiple tools and compensates for the individual weakness, thereby increasing the likelihood of correctly identifying active compounds. Thus, the goal of this project is to evaluate the performance of three docking programs, ICM, NINA, and KarmaDoc, by comparing their individual results to those generated through consensus docking. We hypothesize that consensus docking will outperform individual programs by improving the reliability and accuracy of ligand ranking. First, I'll introduce the methods used in this study. We evaluated three docking programs, ICM, NINA, and KarmaDoc. ICM uses Monte Carlo sampling to globally optimize ligand internal coordinates within the protein pocket, allowing it to explore various poses within the receptor's binding site. The scoring function is the sum of the ICM score and the RTCNN score. NINA also uses Monte Carlo search algorithm to generate ligand poses and incorporates deep learning techniques for pose scoring and prediction. It uses convolutional neural networks, or CNN, to evaluate protein-ligand interactions, producing a CNN score. KarmaDoc integrates deep learning techniques as well, using graph neural networks to encode protein-ligand interactions. Ligand poses are generated in the Euclidean space using EN equivariant networks combined with a mixture density network. KarmaDoc provides three scoring functions: the uncorrected score, the force field or FF score, and the align score. For our purposes, we will use the align score to evaluate performance since it offers the highest reliability in post-prediction. To validate our docking protocol, we docked four well-characterized targets: ABL1 kinase, angiotensin converting enzyme or ACE, cyclooxygenase 2 or COX2, and renin. These targets were chosen for their distinct biological roles and structural diversity, which helps assess the generalizability of our consensus docking approach. This redocking step helped validate the accuracy of our docking protocol and de-risk internal workflow, ensuring the viability of the programs used. Once the protocol was validated, we moved on to docking active and decoy compounds. This approach allowed us to evaluate how well each program could distinguish active compounds from decoys. Ligands were docked using ICM and KarmaDoc, with ICM docking poses rescored using Nina. Each docking tool followed a structured protocol. For ICM, the process involved converting the PDB file into an ICM object, setting up the receptor for docking, positioning the probe and box, preparing the batch of ligands, selecting docking preferences, and running the docking simulation. For Nina, the workflow included inputting protein and ligand structures, setting up the receptor, defining the probe and box, and executing the docking simulation. Similarly, KarmaDoc involved inputting the target protein structure in a PDB format and the ligands in a SMILE and MOL2 formats, and then executing the docking simulation to generate the poses and scores. Default settings were used across all three programs. After obtaining docking results from ICM, Nina, and KarmaDoc, the scores were normalized using min-max normalization. This method rescales the data to a fixed range of 0 to 1, allowing fair comparisons across different targets and enabling the integration of scores for consensus scoring. This step is crucial because each docking program uses a different scoring method, and applying performance metrics directly to raw scores would skew the results. Equal weight was assigned to the three programs, and duplicate scores for each molecule were removed to ensure consistency across the dataset. To evaluate performance, we used two metrics: receiver operating characteristic curves and enrichment factor values. Active and decoy datasets were obtained from the database of useful decoys enhance or DUDE. Active compounds are true binders to the target, while decoys are structurally similar compounds that bind to the target but lack binding activity. The ROC curve plots the true positive rate against the false positive rate. The area under the curve, or AUC, quantifies the performance. An AUC of 0.5 indicates random classification. An AUC greater than 0.5 indicates better than random performance, and an AUC less than 0.5 indicates worse than random. The enrichment factor, or EF, tells us how well a method ranks true positives among the top-ranked compounds. An EF of 1 represents random performance. An EF greater than 1 indicates enrichments of actives, and an EF less than 1 indicates worse than random ranking. To summarize the consensus docking workflow, we validated 4 targets: ABL1 kinase, ACE, COX2, and renin using ICM and KarmaDoc through redocking. After validation, we docked active and decoy datasets using ICM and KarmaDoct and rescored ICM results with Nina. The table shows the total number of compounds along with the number of actives and decoys for each target. We then normalize the scores using min-max normalization to generate consensus scores. Finally, we analyze the data using ROC curves and EF values for the top 1,000 and top 5,000 unique ligands. These metrics allow us to evaluate and compare the performance of each docking program and the consensus approach. Here are the results. First, we validated the ICM docking pipeline using the 4 targets ABL1 kinase, ACE, COX2, and renin. In the images, docked ligands are shown in red while co-crystallized ligands are in cyan. We calculated the root mean square deviation or RMSD to assess docking accuracy. An RMSD below 2 angstrom generally indicates that the docking protocol can accurately reproduce the 3D conformation of the co-crystallized ligand. For ABL1, the RMSD was 0.15 angstrom. For ACE, it was 1.3 angstrom. COX2 had an RMSD of 0.28 angstrom, and renin had a value of 0.22 angstrom. These results show that ICM accurately redocked all 4 crystallized ligands within its binding site. We then validated the KarmaDoc pipeline using the same 4 targets. In this case, docked ligands are shown in yellow and the co-crystallized ligands are shown in blue. For ABL1 kinase, it had an RMSD value of 1.38 angstrom. ACE had a value of 1.58 angstrom. COX2 has a value of 7.16 angstrom, and renin had a value of 3.1 angstrom. While KarmaDoc performed reasonably well for ABL1 kinase and ACE, the 4 targets showed poor prediction of binding poses with incorrect placement of key functional groups as shown in the image. After validating both protocols, we evaluated their ability to distinguish between active and decoy compounds using AUC, area under the curve, and EF, enrichment factor values. Starting with ABL1 kinase, ICM had AUC values of 0.69 for the top 1,000 ligands and 0.74 for the top 5,000 ligands, indicating moderate discrimination between actives and decoys. However, the EF value for the top 0.1% was low, with EF values of 8.5 and 21.5, suggesting poor identification of actives among the top ranked compounds. For ABL1 kinase in Nina, it produced AUC values of 0.76 and 0.69 for the top 1,000 and top 5,000 ligands respectively. While this shows decent discrimination between actives and decoys, enrichment remained low with EF values of 17.2 and 43.5. For ABL1 kinase in KarmaDoc, it achieved higher AUC values of 0.86 and 0.82 in the top 1,000 and top 5,000 ligands respectively, reflecting better discrimination in actives and decoys. However, the EF values for the top 0.1% was 9.7 and 33.8, indicating poor enrichment of actives among the top ranked compounds. For ACE, ICM yield AUC values of 0.72 and 0.79 for the top 1,000 and 5,000 ligands respectively, with EF values of 0 and 17.6. This suggests moderate discrimination but weak enrichment for the top 0.1%. For ACE and NINA, it showed similar AUC values of 0.72 and 0.76 for the top 1,000 ligands and top 5,000 ligands, respectively, with EF values of 8.0 and 22.9 for the top 0.1%, again pointing to modest discrimination between actives and decoys and limited enrichment of actives. For ACE and KarmaDoc, it had lower AUC values of 0.6 and 0.66 with EF values of 16.1 and 21.4 for the top 1,000 ligands and top 5,000 ligands respectively, indicating weaker performance in both discrimination between actives and decoys and enrichment of actives. For COX-2, ICM had a relatively strong AUC value of 0.78 and 0.79 for the top 1,000 ligands and top 5,000 ligands respectively, indicating decent discrimination between actives and decoys. It had EF values of 6.0 and 18.4, reflecting limited enrichment in the top-ranked ligands of the top 0.1%. For COX-2 in NINA, it produced AUC values of 0.69 and 0.74 for the top 1,000 ligands and top 5,000 ligands respectively, with EF values of 6.2 and 16.9, showing similar trends in discrimination between active decoys and enrichments of actives in the top 0.1%. In contrast, COX-2 in KarmaDoc performed near to random for discrimination between actives and decoys, with AUC values of 0.59 and 0.56 in the top 1,000 and top 5,000 ligands respectively, and it had EF values of 0, indicating no meaningful enrichment in the top 0.1%. Finally, for renin, ICM achieved a high AUC value of 0.94 and 0.7 for the top 1,000 and top 5,000 ligands respectively, indicating good discrimination between actives and decoys. The EF values for the top 0.1% were 27.8 and 66.7, showing better enrichment in the top 5,000 ligands, but more limited performance within the top 1,000 ligands. For renin in NINA, it had AUC values of 0.68 and 0.73 for the top 1,000 and top 5,000 ligands respectively, with EF values of 20.8 and 22.7, reflecting moderate discrimination and low enrichment. For renin in KarmaDoc, it also achieved strong AUC values of 0.79 and 0.93 for the top 1,000 and top 5,000 ligands respectively, showing good discrimination between actives and decoys, but it had EF values of 16.7 and 50.5, suggesting weak enrichment in the top 1,000 ligands and close to random enrichment in the top 5,000 ligands. After obtaining docking results from ICM, NINA, and KarmaDoc individually, scores were normalized using the min-max normalization to generate consensus scores. We analyzed the raw curves and enrichment factors for the top 1,000 and top 5,000 ligands across the different combinations of the three programs. This includes ICM and NINA, ICM and KarmaDoc, NINA and KarmaDoc, and all three combined. Starting with ICM and NINA, for ABL1 kinase, the AUC was 0.68 and 0.77 for the top 1,000 and top 5,000 ligands respectively, indicating decent discrimination. However, for the EF values, they were low with values of 10.6 and 30.7 for the top 0.1%, suggesting poor enrichment of actives in the top-ranked compounds. For ACE, AUC values were 0.72 and 0.79 with EF values of 0 and 17.6 for the top 1000 ligands and top 5000 ligands respectively, again showing good discrimination between actives and decoys but poor enrichment. For COX-2, the AUC values were 0.79 and 0.78 for the top 1000 and top 5000 ligands respectively, but the EF values were low with values of 6.0 and 18.4 for the top 0.1%, again showing decent discrimination of actives and decoys and limited enrichment of the actives. Renin showed strong AUC values of 0.94 and 0.7 for the top 1000 and top 5000 ligands respectively, indicating good discrimination between actives and decoys. It had EF values of 27.8 and 66.7, suggesting moderate enrichment in the top 5000 ligands and low enrichment in the top 1000 ligands. Next, for the ICM and Karmadoc combination, for ABL1 kinase, the AUC values were 0.87 and 0.83 for the top 1000 ligands and top 5000 ligands respectively, reflecting strong discrimination. However, enrichment factor values were still low for the top 0.1% of 8.8 and 28.9, suggesting poor enrichment. For ACE, it had AUC values of 0.67 and 0.71 with EF values of 10.8 and 22.7 for the top 1000 and top 5000 ligands respectively, again showing decent discrimination of actives and decoys but poor enrichment. For COX-2, it had AUC values of 0.62 and 0.72 for the top 1000 and top 5000 ligands respectively, with EF values of 0 for the top 0.1%, suggesting decent discrimination and no meaningful enrichment. For renin, it showed good AUC values of 0.87 and 0.8 with EF values of 16.4 and 54.4 in the top 1000 ligands and top 5000 ligands respectively, indicating good performance in the discrimination and weak enrichment in the top 1000 ligands and close to random enrichment in the top 5000 ligands. In the Nina and Karma-Doc combination, for ABL1, the AUC values were 0.79 and 0.76 with EF values of 11.6 and 33.3 for the top 1000 and top 5000 ligands, showing modest discrimination but low enrichment. For ACE, AUC values were 0.71 and 0.78 with EF values of 7.5 and 22.5, again showing decent discrimination but poor enrichment in the top 0.1%. For COX-2, it had AUC values of 0.78 and 0.82 with EF values of 5 and 16.2 for the top 1000 and top 5000 ligands respectively, showing decent discrimination of actives and decoys and limited enrichment for the top 0.1%. For renin, it had AUC values of 0.85 and 0.8 with EF values of 16.1 and 31.3 for the top 1000 and top 5000 ligands respectively, showing decent discrimination of actives and decoys with limited enrichment in the top 0.1%. Finally, for the combination of all three programs, ICM, Nina, and Karma-Doc, ABL1 kinase had AUC values of 0.8 and 0.72 with EF values of 11.6 and 29.8 in the top 1000 ligands and top 5000 ligands respectively, indicating good discrimination between actives and decoys and limited enrichment of active compounds in the top 0.1%. For ACE, it had AUC values of 0.74 and 0.8 with EF values of 6.7 and 21.7 for the top 1,000 and top 5,000 ligands, showing decent discrimination of actives and decoys, but poor enrichment for the top 0.1%. For COX-2, it showed AUC values of 0.83 and 0.78 with EF values of 4.6 and 16.3 for the top 1,000 and top 5,000 ligands respectively, showing good discrimination between actives and decoys with low enrichment for the top 0.1%. For renin, it had AUC values of 0.86 and 0.78 with EF values of 16.4 and 54.4, again suggesting moderate to strong discrimination, but weak enrichment in the top 0.1%. This table summarizes the AUC values and EF values at the top 0.1% for each docking method tested on ABL1 kinase. Among the methods, the ICM-Karma-Dock combination showed the highest AUC value, indicating the strongest performance in discrimination between actives and decoys. Karma-Dock and Nina alone produced the highest EF value, suggesting better ranking of true actives compared to the other methods, although enrichment remained low overall. For ACE, the ICM-Nina-Karma-Dock combination achieved the highest AUC value, indicating the best overall discrimination. The ICM-Karma-Dock pair and Nina alone yield the highest EF value, reflecting better early enrichment compared to other methods, but enrichment still remained low overall. In the case of COX-2, the ICM-Nina-Karma-Dock combination and Nina and Karma-Dock combination had the highest AUC values, suggesting the best overall performance in the discrimination between actives and decoys. ICM alone and ICM and Nina together achieved the highest EF values, suggesting slightly better identification of true actives, although enrichment still remained limited. Finally, for renin, both the ICM-Nina combination and ICM alone performed the best, achieving the highest AUC values and enrichment factors. This indicates strong discrimination between active decoys and relatively better enrichment for renin compared to the other targets. Our results showed that ICM docking algorithm was able to accurately redock the crystallized ligand with RMSD values below 2 angstrom for all 4 targets. In contrast, Karma-Dock produced poor binding interaction predictions across all 4 targets despite having acceptable RMSD values for ABL1 kinase and ACE. Docking actives and decoys remain a standard approach to validating docking protocols, and in this study, we tested all combinations of docking programs. Overall, combining different docking programs generally outperformed individual programs in terms of AUC values and discriminating between actives and decoys. However, enrichment factors, especially in the top 0.1%, remain consistently low across all methods, highlighting the challenge of effectively ranking true actives at the top of the list. Importantly, docking software performance was found to be target dependent. The best performing combination for ABL1 kinase was ICM and Karma-Dock. For ACE, it was ICM-Nina Karma-Dock combination. For COX-2, it was Nina Karma-Dock and the ICM Karma-Dock combination, and for renin it was ICM alone and the ICM Nina combination. These findings highlight the need for further optimization in both post-prediction and scoring, especially when early enrichment of actives is critical for virtual screening applications. Some potential limitations of this study include using a single normalization method, using a single protein structure, and the selection of the dataset. This study relied on only one normalization method, the min-max normalization, and while this approach is commonly used, it is sensitive to outliers, and exploring alternative normalization methods could be beneficial. In particularly, the logistic rank normalization assigns greater weight to top-ranked compounds, which is useful for early drug discovery. Regarding target protein selection, we used a single crystallized structure for each target. However, docking results vary depending on the structural conformation. Ensemble docking could be an approach where each compound is docked using multiple conformers, and this could improve the robustness in future studies. In terms of data selection, the datasets for actives and decoys were sourced from DUDE, which may carry biases, particularly false negatives. To reduce these biases, using the libPCB8 database, which is considered more unbiased, could better replicate real-world scenarios. To improve the accuracy of structure-based virtual screening, future work could explore datametric fusion as a more effective way for combining individual docking scores. These metrics could enhance the ability to distinguish true active compounds from decoys when applied to consensus scoring. In addition, given the target-dependent performance of docking software, another direction is the development of machine learning-based scoring functions tailored to a specific protein target. For instance, in a paper, they demonstrated improved predictive performance using this approach for kinase targets. Additionally, using more docking softwares such as SurfDock could offer further improvements in predicting protein-ligand interactions. With its strong performance in precision, physical plausibility, and practical utility, it offers a valuable tool for advancing molecular interaction studies and structural biology. And finally, with continued optimization of both post-prediction and scoring protocols, as well as addressing the limitations, it could increase the reliability of consensus docking in virtual screening. Finally, thank you for listening, and special thanks to Dr. Matthew Shapira and Conrad Raranso and PCL 472 University of Toronto for their help and guidance throughout the term.